The purpose of this study, carried out by Mattavelli and colleagues, was to clarify the role of the amygdala in processing facial expressions exhibiting different emotions. The amygdalae are small structures in the medial temporal lobe, adjacent to the anterior portion of the hippocampus. And some of the controversy around the amygdala is that it's neither a structural nor functional unit, but rather a bundle of nuclear extensions from other regions, because the amygdala has about 13 nuclei. It is densely interconnected with other structures that synapse on the amygdala for emotion processing, implicit emotional learning, memory, and attention. Previous functional neuroimaging studies have studied patients with amygdala damage and found them to have greater deficits in the perception of fear and danger detection. But converging evidence still arises on whether the amygdala is specific for fear recognition or if it's responsible for emotion recognition in general, since it is the forebrain's most connected structure. This question arose from some previous findings of amygdala activation to facial expressions without any specific effect of emotion type. Previous hypotheses for the role of the amygdala include that it serves as a protection device to detect and avoid danger, and it sends signals to the cortex to avoid danger, such as the motor cortex to execute movements and remove the person from a dangerous situation. A more general hypothesis is that the amygdala detects stimuli in the environment and reacts to them. In other words, the amygdala is involved in a more ambiguous sensation. If it's not clear what the stimulus is in the environment, the amygdala interprets the signal in a vague sense. Due to conflicting previous functional neuroimaging studies, it is unclear the role of the amygdala in emotional processing. Contradictory results have stemmed from the fact that minimal studies have compared many facial expressions within their study, and that different non-emotional stimuli used as the comparison condition were subtracted from the emotion conditions, leading to variability among the literature. The previous study, done by Fitzgerald and colleagues in 2006, measured amygdala responses to the stimuli presented as fearful, disgusted, angry, sad, happy, and neutral facial expressions, compared to the non-face control condition of pictures of radios. Although amygdala activation was present for all facial expressions, its response to fear was higher than for any other emotion, and almost double for anger and disgust. Fitzgerald and colleagues show the significance of implementing the comparison of an emotional face to the neutral face in bridging the gaps of whether amygdala activation is to faces or expressed emotions that previous face versus non-face studies have left. Because of this, Mattavelli and colleagues in this study hypothesized there to be a stronger amygdala activation when processing fearful faces compared to emotional expressions in general, when a low-level baseline is used as a control condition, as compared with control conditions with neutral faces or other pictures, which is a well-formulated hypothesis given the results of previous literature they reviewed. 12 male and 12 female, young and healthy right-handed volunteers of a Western cultural background participated in the experiment, which is quite a small sample size and could lead to a greater chance of low statistical power and an inflated effect size estimation. The researchers implemented a functional magnetic resonance imaging block design with a T2-weighted blood oxygen level dependent contrast to scan amygdala responses to facial expressions of fear, anger, disgust, sadness, happiness, and the 3 comparison conditions. To determine amygdala responses to faces in general, pictures of buildings were used as a non-face contrast. To determine amygdala activations to specific emotions, the basic emotions were contrasted to neutral and mildly happy expressions. Machiavelli and colleagues implemented the 25% mildly happy facial expressions because neutral faces can seem bitter, and the mildly happy faces could therefore be used as a baseline expression. These comparison conditions were appropriate for the question posed on the role of the amygdala to answer whether this brain region is responsive to faces in general rather than an expressed emotion, and if it is liable for perceiving emotions, which emotion it is responsible in detecting. Whole head volumes of the participants were obtained, and its axial slices were positioned accurately for each participant to confirm that imaging was done on the amygdala. After each fMRI experiment, a localizer scan was done to determine the face-selective region of the amygdala. The block design incorporated 5 images of each condition in each block, as can be seen in the figure on the right. Images were shown for 1 second, then a 200 millisecond fixation cross was presented. Blocks were then separated by a 9-second gray screen. Each condition was repeated 5 times in a counterbalanced design. Participants were not explicitly asked which emotions were presented, but rather a red spot detection test was used to monitor attention during the fMRI session. Participants correctly detected a red dot that was superimposed on 18% of the images, showing that they were attentive during the experiment. Then, to determine amygdala activations to the basic emotions and comparison conditions, a block design with 8 conditions of the 5 basic emotions, the building non-face condition, and the mildly happy and neutral face condition was performed. 5 images from each condition were in each block, and the blocks were randomly presented for 1 second, followed by a 200 millisecond fixation cross, then a 9-second fixation cross on a gray screen. Blocks of the 8 conditions were repeated 8 times in a counterbalanced order. The red spot detection task was implemented again to ensure participants' attentiveness. We believe that the block duration should have been longer, since the flashing of images so quickly could have resulted in extraneous variables and activation of brain regions for reasons other than the amygdala's role in detecting fear. After the scans, participants underwent a behavioral task to determine that they could accurately verify the facial expressions. The expression stimuli were shown on a screen, and the participants were asked if they represented fear, anger, disgust, sadness, happiness, or neutral expressions.
So the results were collected using a variety of methods. In the study, the researchers used a post-scan behavioral task to check if the participants in the study were able to correctly categorize the facial expressions into separate emotions that were tested. The hypothesis that the amygdala will produce a strong activation when there is a low-level baseline control used for comparison was confirmed by the results. The participants of the study were able to recognize the different facial emotions for the image set. Figure 2 shows the spatial extent of voxels recorded in the amygdala for each individual emotion being tested. There is significant activation displayed in the left and right hemispheres of the brain for fear, anger, and disgust. However, there is not as much activation for happiness and even less for sadness. Table 1 shows the results of the statistical analysis when comparing the data with the controls, which are images of buildings, 25% happy faces, and neutral faces. The data was also compared on this table using 2 different Harvard-Oxford atlases. Table 1 uses a 5 to 100% amygdala mask for this left side of the table, and a 50 to 100% amygdala mask on the right side of the table. The amygdala masks are used to localize the voxels that are recorded in a spatial arrangement so that the percent signals are accurately recorded. This table confirms that the data collected is from the amygdala region and not from the regions around the amygdala. It also shows that regardless of the control, the data that was collected was consistently higher for fear and anger. Figure 3 shows the face-responsive regions in the left and right amygdala, which is highlighted in green. It uses the functional localizer scan to examine the response to each emotional condition in the face-responsive subregions in the left and the right amygdala. The responses are also defined functionally using the masks from the percent signal change for each of the conditions being tested. The categories that were being tested were the stimuli, which were fearful, angry, disgusted, happy, sad, neutral, 25% happy, and the building control. The experiment also measured the percent signal change of the two hemispheres of the brain. The results of this experiment showed that there was a higher percent signal change for responsiveness upon viewing the face images for fear, anger, disgust, and happiness consistently. Sadness, neutral face images, 25% happiness, and the building images did not show any statistical significance in percent signal change. Now, what were the main takeaways from this investigation? Well, since the main idea of this study was to determine if the amygdala is generally responsive to emotional expression or if it is specific to anger and fear, readers of the paper will learn that the results show that the amygdala is more responsive to fear and anger. However, it is still responsive to other emotions like happiness. Through different testing that have been already mentioned, the researchers were able to compare the results with other studies such as works from Sergey and Fitzgerald. In Fitzgerald's research study, the results the researchers found led them to conclude that the amygdala is responsible for the perception of emotion. However, this conclusion is faulty. Specifically, this conclusion is believed to be misinterpreted because the data does not account for perception, and rather it accounts only for a response. What did the study tell us about the brain and cognition/behavior that we didn't already know before? The goal of this study was to determine whether the amygdala is responsive to emotional expressions in general, or if it is selective for particular expressions that signal the presence of the potential threat. The results of this study furthered evolutionary thinking within the field of cognitive neuroscience by showing that different procedural choices can have a substantial effect on the pattern of amygdala activation. This helps resolve previous controversies on the role of this brain region. Just by learning this much about the amygdala allows researchers to further investigate different therapies for mood disorders and also further research about the amygdala to better understand its function and purpose. This study can also be used to advance medicine if further research is conducted regarding the association of the amygdala and certain mood disorders under the gaze of this study. Also, since the amygdala is responsible for eating, sexual function, and aggression, the research could be used to further the study in ways that can improve the disorders of those three things. For example, with eating disorders, the amygdala can be studied further to help improve treatment of certain disorders.